Hello dear students, hope all of you are at home and you are doing well. I am Əyyubova from Department of Histology, Embryology and Cytology. Today we start lectures on general histology. Today we will focus on histology as biological and medical science, tissues definition and classification. Epithelial tissues will be our main focus today. We will talk about its histogenesis, classification, morphofunctional features, then the covering epithelium. Glandular epithelia, the mechanism of periods of secretion will be our further discussion. As we know, the histos, it means tissue, and histology, that is a science which studies tissues of the human body. As we know, 200 types of cells that compose the human body, they are arranged in tissues. The tissues, each of them, it has its own characteristics and functions. Tissues in turn form organs. They are assembled in more complex structural and functional organizations. They are the organs. Organs will form system organs, and system organs together compose whole body. As we know, all of the cells of the body develop from just one cell. That is the result of fertilization, that is the zygote. The zygote undergo steps of differentiation, specialization, proliferation. Finally, it leads to formation of the blastula. From the blastula will develop germ layers, and further, each of these germ layers will differentiate into some groups of tissue types. Initially, we have totipotential embryonic stem cell, that is the zygote. The zygote, as we know, differentiates, subdivides into pluripotential embryonic stem cells. Further, their division gives rise to multipotential stem cells. After their division, we will have unipotential, and each of the unipotential stem cell will develop into just one cell type. Now let's talk how the cells undergo differentiation and how they specialize to become final definitive cells. The specialization each of the cell type is realized in the process of cell differentiation. And differentiation of cells is a stable structural and functional transformation of cells into various specialized cells. If we will compare the determination with the differentiation, here you can see the characteristics. Actually, the determination, it is the process by which portions of the genome are selected for expression in different embryonic cells. So different embryonic cells will express different genes, and it occurs mainly in totipotential embryonic stem cells. These are cells which are able to differentiate into all of the cell types, not just of the embryo, but also extra embryonic organs. If you remember, we studied this topic in the first lesson. And in first lecture, you listened about it. A result of asymmetric segregation of cytoplasmic determinants, and it's responsible for assigning the fate of the cells. But the differentiation of cells is due to their determination. Determination is a process of determining the path, direction, program for the development of material of embryonic primordia with the formation of specialized cells. The mechanism of determination is associated with persistent change in the processes of repression, blocking, and the expression, activation of various genes. Here we can see how the determined cell, it is determined by its genome, then will differentiate in different ways. Finally, it will give rise to different cell types. And here The differentiation follows determination as the cell elaborates a cell-specific developmental program. Differentiation results in the presence of cell types that have clear-cut identities, such as muscle cells, nerve cells, epithelial cells, and cells of skin, for example. What is the base of these processes? Actually, we have to know that it should be understood that the genome contains two kinds of genes. First of all, housekeeping genes. These genes are necessary to maintain the vital functions of the body, which are expressed particularly in all tissues and cells. These functions include protection, for example, from the environment, nutrient acquisition, communication, movement, renewal of senescent molecules, molecular catabolism, and energy generation. And in addition, there are specific genes, the activation of which leads to cell differentiation. So we are coming to the study of tissues and how tissues are combined in various ways to form organs and membranes of the human body. As we know, the human body contains four primary types of tissues, that is epithelial, connective, muscle, and neural tissue. Each tissue type, it includes two main components. These are cells and extracellular matrix components. The extracellular matrix include fibers and ground substance, which contain organic and inorganic molecules which are bound to water. Today, we will focus on epithelial tissue. The epithelial cells will form epithelial tissue. Actually, epithelial tissue, it is covering tissue of the human body, and it will cover its outer surface and its inner surfaces, and also it will form our glands. The epithelial tissue may develop from different sources. First of all, ectoderm. It gives rise to the epidermis of the skin, lining of the mouth and nasal cavity, cornea, sweet and sebaceous glands, and mammary glands. The endoderm gives rise to the lining of the gastrointestinal and respiratory systems and to glands of the gastrointestinal system. Then the mesoderm, it will give rise to, if we are talking about the intermediate mesoderm, it will develop into uriniferous tubules of the kidney, then the lining of the reproductive systems. If we are talking about the lining of the blood vessels, circulatory system, they will develop from the mesenchyme, and the lining of the body cavities, the mesothelium, that is the simple squamous epithelium, that will develop from the visceral, from the lateral mesoderm, from its layers. So, we have to know that the epithelial tissue, it performs different functions. It is protection, secretion, absorption, then transcellular transport of substances, of molecules across the epithelial sheets. Also, we have to know that inside of the epithelial tissue, we will have nerve endings, so that detection of sensations, that is, for example, taste, hearing, and so on, that is also functioning. It is a receptor function of the epithelium. Then, if we will talk about its general classification, here we can see that epithelium, it is covering epithelium. The first of our focus will be on the covering epithelium. Its classification, we can see here that is classified into simple and stratified epithelia. The simple epithelia also subdivided into squamous, cuboidal, columnar, pseudostratified. It depends on the shape of cells. Then we will have ciliated and non-ciliated, and stratified epithelia also of different types. Each of them we will discuss today. But before going to the each of the tissue type, each of the epithelial tissue type, let's talk about the characteristics which differ the epithelial tissue from the other tissue types of the human body. The first, here you can see that the cells of the epithelial tissue, between them there are not any blood vessels, that is avascular tissue, and it means that between the cells we cannot see blood vessels. That's why nourishment of the epithelial cells is provided from the underlying connective tissue blood vessels. Those nutritive substances and oxygen will diffuse through the tissue, connective tissue, and will reach epithelial cells. Then epithelial tissue cells, they form sheets of cells. They are tightly joined each other. It means that between them there are almost not extracellular space and extracellular matrix. By joining to each other, they form sheets of cells. So the sheets of cells will form the epithelial tissue. Then other characteristic of the tissue, it is the polarity. And polarity, it means that in epithelial tissue, the cells will have different morphofunctional and biochemical domains. That's why in most of the epithelial cells, we will distinguish two parts, the apical part and the basal part. They will have different also content. Then what we have to know, it is the epithelial cells, epithelial tissue, separated from the underlying connective tissue by the basement membrane. So here we will see the membrane which separate the epithelial tissue from the underlying connective tissue. Now let's look at the structure of the basement membrane under the electron microscope. We can see that the basement membrane, it includes two components, that is a basal lamina and the reticular lamina. The basal lamina includes one layer that is electron-lucent and the second one electron-dense layer. Here you can see two epithelial cells, one of them here, the other one is here. Between them we can see interdigitations. These are projections of neighbor cells that interdigitate with each other. Now let's talk about each of the type of epithelia which we can find in different parts of the human body. The first type here, that is the simple squamous. Actually, we have to know that epithelia, covering epithelia, they are classified according to number of cell layers from basal lamina, basement membrane, till free surface of the epithelium. So that we have simple epithelia, which include just one layer of cells that rest on the basal lamina, and the stratified, which includes several layers of cells. First of all, simple squamous epithelium. Simple squamous, it includes one layer of flattened cells. Inside of them we can see nuclei which also have flattened shape. These epithelia we can find in the lining of the endothelium of the blood vessels, then the lining of the alveoli of the lungs, then in the Henle's loop of the kidney tubules, and also in the serous membranes which cover the peritoneal cavity, then the pleural cavity. That epithelium is called mesothelium. Now we will look at the illustration of the blood capillary. For example, pay attention to the endothelials, that is the longitudinal section of the blood vessel, and innermost coat here, that is endothelium, that covers inner surface of the blood vessel. Simple columnar epithelium, it includes one layer of columnar or cylindrical shaped cells. That epithelium we can find in most of the digestive system, in gallbladder, also in small bronchi, but in different parts of the body. On the surface of the epithelial cells here, we can see different modifications. For example, microvilli, which present on the apical surface of these cells, will form striated border. that is characteristic of the alimentary canal. But in other places we will find cilia instead of the microvilli, and that will be characteristic for epididymis, ductal efferents of the testes. Then we will find in the uterus, in oviducts, then in small bronchi, then so that will be the simple columnar epithelium, which will carry on its surface cilia. Now let's look here, simple columnar, and with microvilli, it will perform absorption. If it will carry cilia, it will be movement of mucus or X, depending on the location. And here we also can see where it is found. Then now look at them. We have also simple columnar, simple cuboidal epithelium. Simple cuboidal epithelium, it is next type, that includes just one layer of cuboidal shaped cells which rest on the basal lamina. That is kidney tubules. Here on the example of kidney tubules we can see the simple cuboidal epithelium. This one also we can find in ducts of many glands covering the ovary, then from the kidney tubules. The main function of this epithelium is secretion, absorption, and protection. Now let's go to pseudostratified columnar. Besides simple and conventional stratified epithelia, we also have pseudostratified epithelia. That is, include just one layer of cells which rest on the basal lamina, but it is appeared like stratified. The key point here is that the cells have different shapes. That's why the nuclei will located in different levels. Some cells will have broad base, broad apex, and narrow base. Others will have broad base and narrow apex. That's why they are not in the same in the shape. That's why they will form different levels of nuclei, which will give us the impression of stratified epithelia. The pseudostratified epithelia we can find in the lining of the trachea, primary bronchi, then epididymis and ductus deferens, then auditory tube, part of tympanic cavity, nasal cavity, lacrimal sac, male urethra, and large excretor ducts. What we have to know here that in most of those epithelia, on the surface of these epithelial cells, we will have cilia. That's why it will also have name ciliated epithelium, pseudostratified ciliated epithelium. If we will look at it under the in histological section, we can see in micrograph the appearance of the pseudostratified epithelia. Pay attention that on the basal lamina we will see different levels of nuclei. That will be the pseudostratified epithelia. Now we are coming to transitional epithelia. Transitional epithelia, it is like transition. It is because the shape of the epithelium changes depending on the state of the organ. If this one actually the transition epithelium we can find in the urinary system, beginning from the renal calyces until urethra. In here we can see that this one composed of also here that is cells which form several layers. So that's what it is. The transition epithelium, it is type of stratified epithelium, and on the surface we can see dome-shaped cells. In most cases they are binucleated. When the organ is empty, here we can see several layers of cells, but when organ is stretched, the cells also become squamous, flattened, and here we will distinguish just 2 or 3 layers of cells. So that is the transitional epithelium. It is a type of stratified epithelia. Now let's look at the stratified squamous epithelium. stratified it because it includes several layers of cells, and squamous it is because of the shape of most superficial layer. So in stratified epithelium we will talk about the shape of the epithelia according to shape of cells in the most superficial layer. That is a type of stratified. It is stratified squamous non-keratinized epithelium. It includes several layers of cells, and in most superficial layer we can see flattened cells which include nuclei. This type of epithelium we can find in lining of mouth, epiglottis, esophagus, vocal folds, and vagina. Its function is protection and secretion. Let's look at this epithelium in histological section. Different layers of cells, and in the surface of the layer we can see the nuclei inside of the cells. Now let's look at the stratified keratinized epithelium. In stratified keratinized epithelium, pay attention that it also stratified because it includes several layers of cells, but here the superficial layer includes dead cells. They are horns actually, and we cannot distinguish even individual cells because they form complete layer. Inside of these cells, the cytoplasm and nucleus, they are replaced by keratin filaments. That one we can find in epidermis. It covers the epidermis of the skin. So it also includes several layers. Now let's come to the glandular epithelium. The glandular epithelium, it forms glands. And during the development of certain regions of the body, epithelial cells invade the underlying connective tissue, and they will form here the glands. The epithelial cells here will form parenchyma of the gland, and it will be surrounded by the basal lamina, and they will form the glands of the body. The surrounding connective tissue will form stroma, and it will support the parenchyma of the gland by providing vascular and neural supplies, and its structural elements will form capsules, and it will cover the entire gland. From the capsule will arise septa, which will subdivide the gland into lobes and lobules. Actually, there are two principal types of glands, that are the exocrine glands, which have ducts, and through these ducts the secretory products are delivered onto the epithelial surface. And we have endocrine glands, or they are ductless, and consequently the secretory product is delivered directly into the bloodstream because they have very rich blood supply. So that are the two main kinds of glands. The endocrine glands you will study on next semester. Today we will focus on mainly exocrine glands. What is the classification of the exocrine glands? These glands are classified according to different features. Depending on the number of cells which form the gland, they are classified into unicellular, if the single cell is the entire gland, for example, goblet cell, or multicellular, if the gland is composed of more than just a single cell. Then additional classifications are based on the type of secretion the gland produces. We have to distinguish serous. It is watery. For example, they produce mainly protein components. For example, parotid gland. Then mucus, if they produce viscous mucus, for example, in minor salivary glands of the palate. Then mixed, serous and mucus, both of them. And we have also sebaceous glands which produce sebum or fat. And other classifications are also based on the shape of the secretory portions and the duct portions. Actually, if it is the exocrine gland, it has secretory portion and the duct. So according to the shape of the secretor portions, we can distinguish acinar glands. Pay attention if they have acinus shape, the secretor portion, and if it has tubulars, that will be tubular glands. And also multicellular glands are classified as simple if they have just one duct and their duct do not branch. Pay attention, all of them are simple glands. They are ducts, just one, they do not branch, and they will be compound if their ducts do branch. They are further categorized according to the morphology of the secretor units, and here you can see tubular, acinar gland, or if they have both of them, it will be tubular alveolar glands. According to branching of the secretor portions, we will distinguish unbranched, just one secretor portion, but if it is branched, the secretor portion is branched, that will be branched gland. For example, this will be simple tubular gland, that will be coiled because of the secretor portion is coiling here. That will be simple tubular gland, but it will be branched because secretor portion is branched. In this case, we are looking at the compound glands. That will be compound tubular and branched. That will be compound alveolar. It is also branched. That will be tubular alveolar because both of types of secretor portions we can see here. It will be compound, branched, tubular alveolar gland. If we will look at the other, pay attention to this illustration, that is unicellular gland, that is the goblet cell, which is mucus production cell. And here we also can see multicellular glands. Each of them produce mucus, for example. These are the simple and these are compound glands. If we will look at the mode of secretion, we can distinguish here merocrine, then apocrine and holocrine types of secretion. In merocrine, the cell, when it releases secret, it releases via exocytosis, and the cell do not destroy it. Its structural integrity is not destroyed, and that is neat and clear type of secretion. It is characteristic to many types of glands of the human body, for example, salivary glands. In this type, that is apocrine type of secretion, when the secret releasing, on that case, the apical portion of the cell is break up and it also will be released together with secret. Then the cell will restore itself by regeneration. That is, for example, in mammary gland, in some of sweet glands. And the holocrine type of secretion, when the cell produces its secret, it stores, then during secretion, all of the content of the cell is released outside of the cell. Now, if we will look at the unicellular gland, that is the goblet cell here, pay attention, it actually has columnar shape, but when it produces secret, it stores the secret in secretor vesicles, and they contain mucinogen. They fill the apical portion of the cell and distort this portion. That's why you will see it as in the bulged shaped apical part of the cell. That is the goblet cell, which is mucus production cell. That one we can find in the lining of respiratory system and the digestive system. But we should take in mind that we have endocrine cells. These are individual cells which scattered among the epithelial cells lining the digestive tract and respiratory system. These particular endocrine cells represent the diffuse neuroendocrine system or DNES system. Certain paracrine and endocrine hormones are products of these DNES cells, and because they are colored by argentum nitrates, that's why they called argentaphilic or argyrophilic cells. They we can also find in the respiratory system. Here you are looking how the secret is released. As I mentioned, that is merocrine, apocrine, and holocrine type of secretions. Now let's come to the surface modifications of the cells. As we know, the epithelial cells are polarized cells. That's why they have apical domain, pay attention, apical domain, and basolateral domain. Here you can see the schematic representation of the epithelial cells, neighbor epithelial cells, one cell here, second one, and the third one. Here you can see the electron microscope representation of lateral domain of the cell. The basolateral domain we can subdivide into the lateral domains and the basal domain. Because these regions are different functionally, each may have surface modifications and specializations related to their functions. For example, the apical surface of many epithelial cells possess microvilli, as you can see here, or it may have cilia. And the basolateral regions will have junctional specializations and also intercellular interdigitations. We will see in more basal regions. The apical and basolateral domains are separated from each other via tight junctions. Pay attention, these are regions of tight junctions or zonal occludence. That tight junction will isolate the apical domain from the basolateral domain. In apical domain, in apical cell membrane, we can find different ion channels, carrier proteins, then pumps. Also receptor proteins we will have there, and also these projections, different types of projections. These are like microvilli, stereocilia, cilia, or flagella. The structure of them we studied in the course of cytology, that's why I will not stop here. Now let's go to the lateral specializations of the cell, of the epithelial cell. In lateral surfaces, in lateral cell membranes, there are located specific intercellular junctional complexes. Here we can distinguish three types of intercellular junctions. Let's look at the next illustration. So these are continuous epithelial cells. They held together via these intercellular junctional complexes. Here we can distinguish occluding junctions, which locate more apically, then anchoring junctions, which located basally, near to the basal part. Then we have communicating junctions. Now we will focus on each of these type of the junctional complexes. Zonal occludence, it is the mostly apically located intercellular junctional complex, and it is formed by fusion of the outer leaflets of adjacent cell membranes. The fusion extends along the entire circumference of epithelial cells, and when it is viewed with freeze fracture electron microscopy, fusion, pay attention, if we will look at the freeze fracture electron microscopy, fusion strands, linear arrangements of transmembrane proteins are evident on the P phase, and according to this one, linear grooves are present in E phase. That is the occluding junction. Depending on the integrity of the tight junction, several substances will be actually zonal occludence. It occlude the intercellular space. It is very important in the organs like urinary bladder, intestines. They will occlude intercellular space and prevent passage of ions, then water between the cells into the intercellular space. It is very important. But in some organs, it may be permeable for some components. What are the transmembrane proteins which will form the occluding junctions? Here, the transmembrane proteins in the membranes of both cells, they contact each other in a calcium-dependent manner, and they obliterate that space. There are three types of transmembrane proteins, and they present in tight junctions. They are claudins. They are most important of the other components. Then we have occludins and junctional adhesion molecules. The claudins here, they are main type of the intercellular junctional protein. It will occlude intercellular space. So they will form belt-like junction that will encircle the entire circumference of the cell. Below, we will see adherence junctions. They are of two types. It will be zonular adherence and the desmosome. Now we will look at them. So let's look at the zonular adherence. In zonular adherence, we will see, pay attention to its structure. Here we can see adjoining cell membranes of one cell, that is membrane of the other cell. Inside of the zonular adherence, we can see other kinds of the transmembrane proteins. And zonular adherence, like zonular occludens, it is belt-like structure which go around all of the circumference of the cell, and it's located just basal to the zonular occludens and also to encircle the cell. The intercellular space here of 15 to 20 nanometers between the outer leaflets of the adjacent cell membranes and is occupied by the extracellular moieties of catechols. Pay attention, this is catechols. You remember catechols belong to cell adhesion receptors. These are glycoproteins which present inside of the cell membranes. And if you remember, catechols, these are calcium-dependent transmembrane proteins which will join each other in the presence of calcium ions. Then catenin will bind to the other proteins that will be catenin, vinculin, and by these proteins, the catenin will bind to actin filaments. Actin filaments will go around all of the circumference along the circumference of the cell, parallel to the cell membrane, and the actin filaments bind to each other via alpha-actinin protein, so that it will be the zonal adherence. And here, in the presence of calcium, the hinge regions of the calcium of the catenins will join each other. Here we have to mention that the presence of cytoskeleton elements under the cell membranes of adjoining cells impart mechanical strength to these type of junctions. So adherence junctions, these are not just a binding of cell membranes of two neighbor cells. It's also adjoining of the cytoskeleton elements. That's why these types of junctions will provide mechanical strength to intercellular junction. They will be present in the tissues and organs where this is needed, that are always undergo abrasion, friction, and so on. It's especially in much amount we will see in epithelial lining of the internal organs or in surface lining of the body. So after the zonular adherence, we are going to the next type of the junctional complex. It will be the desmosome. Desmosome, it's also type of the adherence junctions. Here under the cell membrane, we will see desmosomal plaques. So the desmosome, these are the last of the other components, and here we can see that is the spot weld-like junctions, and they are include these disc-shaped regions, which include two plaques, attachment plaques, the inner and outer dense plaques, and they located opposite each other on the cytoplasmic aspects of the plasma membranes. The extracellular space between two cells is approximately 30 nanometers in width, and it is occupied by the two types of transmembrane proteins, which called desmocollin and desmoglein. And they also belong to cadherin family of proteins. They also calcium-dependent adhesion molecules. In the presence of calcium ions, the extracellular portions of these proteins contact each other and form bonds that attach these adjacent cells to one another. Also, we have to know that outer dense flux is closely adhering to the cytoplasmic aspect of the plasmalemma. And inside of this flux, we can find, pay attention, inside of this flux, we can find the glycoproteins. They are the plakophilins and plakoglobins, and they are held together via desmoplakin proteins. The intracellular part of the desmocollins and desmogleins contact and stabilized by these plakoglobins and plakophilins. To this end, if you can see here, from the cytoplasmic part to this desmosomal flux come intermediate filaments, and they insert here, then they form hairpin turn, and then return to the cytoplasm in both sides. So intermediate filaments will appear in electron micrograph like also flux, but they will called inner dense flux. So in the desmosome, we will see both of these flux types. And finally, we came to the third type of intercellular junctional complexes, that are the gap junctions. The gap junctions, these are mostly abundant type of the junctional complexes, and they present in epithelial tissues, in neurons, cardiac and smooth muscle cells. Gap junctions, they provide intercellular communication because they permit small molecules to pass through the narrow, wide intercellular space. The gap junctions couple cells chemically and electrically to each other, but that way facilitate intercellular communication not just in adult and also in embryonic tissues. If we are looking at the structure of gap junction, we can see here that it is formed by the transmembrane proteins which called connexins. 6 connexins, these transmembrane channel-forming proteins, gather to form aqueous channels known as connexons. One connexon inside of one cell and the connexon of the neighbor cell, they come align with each other and they form the gap junction. The number of connexons that are present in a gap junction varies from a few to several thousands. Connexons of one side of the gap junction that are in register with connexons on the opposing side of the gap junction, they will form hydrophilic communication channel, and this channel is 1.5 to 2 nanometers in diameter, and they will provide molecules with less than 1 kilodalton in size to pass between adjoining cells. It will be mainly ions and in small amount water molecules. When the gap junction will be closed and when it will be open. Actually, it is known that the increase in cytosolic calcium concentration or a decrease in cytosolic pH will close gap junction, but cytoplasmic, if pH is high or calcium is low, it will open gap junction. Finally, we told about all of the junctional complexes. That was our tight junction, that is adherence junctions, the zonular adherence, and the desmosomes. Finally, we told about the gap junctions. Now let's go to specializations of the basal surface, basal membrane, cell membrane of the epithelial cells. Basal lamina, it will, you know that basal cell membrane will attach to the basement membrane. Here you can see the basement membrane. For that one, it will have the hemidesmosomes, then focal adhesions. Focal adhesions you studied in course of cytology. And also there will be the enfoldings, basal membrane enfoldings. They will form plications here. These applications, especially those concerned in cells which concerned in the ion transport, they will increase cell membrane surface area, and it will compartmentalize the basal cytoplasm into mitochondria housing segments. This presence of mitochondria together with plicated cell membrane will make the cell to appear striated when you will view by light microscopy. Then hemidesmosomes. Hemidesmosomes actually they appear as half of a desmosome and are located on the basal cell membrane. We have two kinds of hemidesmosomes, first type and the second type. The first type, it is present mainly in stratified epithelia, stratified squamous, then transitional, and the second type present mainly in simple columnar epithelium. Let's look at the structure of hemidesmosome. Hemidesmosome, it includes one plaque, as hemi it implies that it's like half of the desmosome. In these plaques, we can find the proteins lectin, then bullous pemphigus 230, then erbil proteins, pay attention, they form this plaque. Into this plaque from the cytoplasmic site will insert intermediate filaments like in the desmosomes. So the hemidesmosome is also will be strengthened by the presence of the cytoskeleton elements. Here in this case, these are also intermediate filaments. If we will look at the membrane part of the hemidesmosome, here we can see the transmembrane proteins, mainly integrins. Also type 17 collagen or bullous pemphigus protein 115. These transmembrane proteins in one side they insert into this plaque, and in extracellular site they insert into the basement membrane. By that way they provide attachment of the epithelial cell to the basement membrane. By that way to the connective tissue, underlying connective tissue. These proteins will bind to basement membrane components. These are laminin proteins and collagen of fourth type collagen, fourth type, which is present inside of the basement membrane. The structure of the basement membrane I will explain to you on the next lecture. That is the end of our lecture today, dear students. Thank you for your attention.